else left the room apart from you guys, did they? We're going to start early, why not? Am I going? Oh, yeah. Okay, start early and hopefully I won't run over time for a change. So I'm trying to fit in, I'm aiming to try and fit in 2 case studies here on bacterial diseases. Should be able to do that. So the first is common scab, which, as we mentioned before, is a kind of a superficial disease that just makes things pretty ugly because no one buys potatoes that kind of look with like that with these dark scabby bits on them. So that's the problem. You get ugly potatoes no one wants to buy, and in the processing sector, if it's a deep lesion, even though they peel them occasionally, they can't peel them deep enough, and so they'll get things like this, and so you will occasionally find chips that look pretty ugly, and then people get fairly upset when they see black bits on their chips. To the extent that the processing companies, I mean, Tasmania is primarily a processing potato state, and yet common scab was still regarded as a major problem, even though most of it's peeled off, because that is unacceptable by McDonald's, which were the major consumer of Tasmanian potatoes. You can't have that in their little box of fries. So any crop that had high levels of common scab was just rejected. Was at too high a level, was just, I wouldn't buy it, and so it'd be a complete loss. So a superficial disease causing no yield loss can have a major economic loss. It's called common scab because, hey, guess what, it's common and it occurs pretty well globally. It's actually regarded as really, after late blight, globally, common scab is pretty well up there in terms of one of the most important diseases globally, and again, because of cosmetic things. In Tassie, it's about, in Australia, on the processing sector, not fresh market, it's much higher in fresh market. Just in the processing sector, it's about a $2 million a year issue. The fresh market, I suspect it's much more. And in the seed industry as well. The seed industry, they'll reject crops based on the presence of that. So it's probably closer to 5. What causes the disease? It's actually caused by an actinomycetes, or a gram-positive filamentous bacteria of the genus Streptomyces. So Streptomyces that produce a whole bunch of our antibiotics and other things. These can cause plant diseases. The type species is Streptomyces scabies, or sometimes called Streptomyces scabii. But what makes it particularly weird is that there are a large number of, a dozen or so different species causing common scab around the world, including Tasmania. We have at least a couple of species here, although it's not been particularly well characterised. Why? Little aside. The reason is, hang on, do I come up later? I think I do. I'll talk about it later. So there's multiple species involved. Most of them are Streptomyces scabies, but there are other species. Why is that important? Because you get variability in virulence between different strains and different species, which might be why some regions are particularly worse than others. But also, some of the native species that become pathogenic can be better adapted. So it might be better adapted for a particular soil type. So Streptomyces scabies, the type member, doesn't like acid conditions, tends to grow best about pH 7, but there are native versions that can be quite acid tolerant and cause disease in acid soils. So that becomes a problem. For us, detection and diagnosis is really, really tough because they all look the same. We've got Streptomyces scabies that are pathogens, we've got Streptomyces scabies that are non-pathogens. They are morphologically identical. If you make a serological assay, it won't be able to differentiate it. Even using a molecular assay on things like the standard reference genes like the ribosomal genes, you won't be able to tell the difference. You have to target the factor that actually causes disease. So with these particular pathogens, there is something called a pathogenicity island. This is a piece of DNA in the chromosome, a fairly big one, it's about 50 kb, with lots and lots and lots and lots and lots and lots of genes on it, which seems to be a bit of a garbage heap. So this bacteria accumulates pathogenicity type genes and stuffs them into this piece of DNA. In there happens to be several genes that are responsible for biosynthesis of a toxin. And this toxin is what makes it pathogenic. So that toxin actually causes the infection and allows it to happen. So this piece of nucleic acid with all these genes can mobilize. So it can actually move between species. So it can actually go from our scabies into our native species by conjugation and turn the native species into pathogens. It can also be lost, which is why some scabies are no longer pathogens, because they've lost the ability to produce the toxin. That's why we have this variation, which makes it tricky for diagnosis. Anyway, we also get highly variable symptoms. Now typically, and that's not a great example, but typically you get these star-shaped sort of lesions, fairly superficial. But you can get these raised ones that actually stick out 2 or 3 millimeters. You can also get, and that's not the worst one, these sunken lesions. I've seen some where there's almost no potato left because it's actually been incredibly aggressive. And in that instance there was yield loss where it actually has, penetrated. So typically roundish or star-shaped, cracked, torn edges, can be raised, can be sunken up to several millimeters deep. And part of that depends on how well the plant defends it. I said before, the scab is actually the plant stopping penetration of the infection. So the infection occurs and it seals it off. If the pathogen happens to be quite aggressive and manages to break through that, it'll do another one, and then it might do another one. And that's where you get your deep scabbing, is because it's managed to crack its way through 1 or 2 layers of defence, and then eventually it gets held up and doesn't go any further. You can, if it's really horrible, end up with the entire tuber covered with lesions. Diagnosis as well is really tricky because that is not common scab. That is powdery scab, and they look pretty similar. Why do we care? Why do we want to know the difference? Because powdery scab is caused by a completely different organism that is completely that works under completely different environmental conditions. So, Spongospora subterranea, it's actually a protozoan pathogen. It's promoted by cool, wet soil conditions and irrigation during tuber elongation. Common scab likes warm, dry soil conditions, and irrigation is a great control, so completely the opposite. So you've got to know what you're dealing with, otherwise your control strategy will be reversed. In fact, in Tasmania now, common scab used to be a much bigger problem than it is now. We now have a more robust irrigation system to help control common scab, but also to get better tuber uniformity. And guess what? Powdery scab is now our biggest issue because we're putting more water around. Anyway, let's look at the pathogenicity cycle. So it needs to survive, it needs to move, it needs to infect, it needs to develop disease. Survival. This pathogen, Streptomyces, is extremely efficient as a saprophyte. It can survive in the soil, particularly associated with organic matter, and survive there for decades. In fact, they've gone into should be virgin soils, but probably soils that they've cleared the forests, so they've run some cattle on it, then they've grown potatoes and got nailed with common scab. They go, how did that happen? Probably because those cattle were fed infected tubers, but anyway, and contaminated the soil. But it will survive. It'll actually love rotations that involve pasture, because pasture has high organic matter, and the pathogen will actually grow really well in that organic matter saprophytically, waiting for a potato to come along and then give you nasty disease. Pathogenesis obviously is favoured by its favoured host. So you grow potatoes and the proportion of pathogenic strains will go up and the proportion of non-pathogenic strains will go down. With rotation, the pathogenic strains will probably even out, but will never go away because they are incredibly efficient saprophytes. It also survives by associating with seed tubers. So we saw our infected tubers with lesions all over them. They're containing inoculum. The inoculum's sitting in those lesions. It's also on coating the outside of the tuber that perhaps doesn't show any symptoms. And they will survive there quite happily until you plant that again. So for the seed industry, the seed tuber industry, they plant tubers, they don't plant true seeds. If you have a crop that's got, I think at the moment it's 2% of tubers having any lesions at all, you will lose certification. They won't be able to be sold as certified seed, because we know if you plant a tuber with a lesion on it, you will get common scab from that tuber on the subsequent plants, given the right environmental conditions. So it survives in the soil very well, it survives on tubers, and obviously moves on tubers too. So movement from region to region is primarily on seed tubers. So it's gone around the world, and it's gone around the world primarily because in the early days we sent potatoes around the world, and all the potatoes had some lesions on them, and then that's happened. And then the pathogen, the original Streptomyces scabies, has got into the soil, has mated with the native species, and has created a whole bunch of new species in all the different regions it visits. So one of the commonalities around the world is Streptomyces scabies. In the individual regions you start getting some unique differences. So, movement of seed tubers is a major way of the pathogen spreading. It lives in the soil very efficiently, so anything that moves the soil around will also spread the pathogen. So soil adhering to vehicles, blowing around, cultural operations, soil moisture, anything, water washing soil down. Got soil insects in there too that can carry things around, but can also actually help with infection processes. Animal waste, I think this is much bigger than we tend to give it credit for as well. This pathogen is very, very tough. As I said, it's a great survivor. It'll survive in soil. You can air dry the soil and then it'll still be there. It'll survive for decades in the soil, but it'll also survive passage through animal guts. So again, think about our conditions here. We've got a big processing sector. We get a bunch of contaminated tubers. We peel them all off, peel all the scabby stuff off it to make our nice French fries. And what do we do with the scabby stuff? We might try and recover some of that and make it into hash browns, whatever else. But a lot of it ends up being, we don't want to just dump it. So they actually, it's a good food source. So they feed it to dairy cattle. So scabby, concentrated scabby potato material gets fed to dairy cattle, has the passage quite nicely through the cattle gut and gets deposited in a lovely huge lump of organic matter and persists that way. So again, a really useful way for the pathogen to spread around. So overwintering dissemination. Now infection. Infection, as I said, these bacteria, they can't force their way in. This might be a possible exception. There is some evidence to suggest that this guy, and remember this guy looks a bit like a fungus. It's got these hyphal-like structures and it grows across an agar plate a bit like a fungus as opposed to a bacterial colony. There is some microscopic evidence to suggest that maybe it can punch its way into a cell. But most of the literature you read, and really most of the evidence is that it actually goes in through natural openings. So in the tuber, it doesn't have stomata, but it has these little dark spots. They're lenticels. So these lenticels are actually breathing holes. So that's essentially an underground stem, a swollen underground stem is a tuber, and it needs to breathe, it needs to respire. So all of these little holes, these little lenticels, actually allow for gaseous exchange. They're not completely open like stomata, so they're filled full of what we call packing cells, these loose cells in the lenticel, but that actually allows gas exchange. But they're also relatively open, and that actually allows the pathogen to enter through past all the waxy defences on the skin surface into the internal part of the tuber. Who's seen who's ever grown potatoes when it's been quite waterlogged or quite wet, and you see all the white fluffy spots on the tubers? Has anyone seen that? That's lenticels proliferating. So here you've got these. As I said, we have these lenticels, and they're filled full of loose cells. Under high humidity or moist conditions, these replicate like crazy, and they actually stick out through the surface of the tuber and look white, fluffy, and then eventually go brown and horrible, and that can be a problem if you're trying to sell them as well. Infection. Environmental conditions. If we look at our disease triangle again, we've got our pathogen, we've got our host. Environmental conditions play a really important part in this pathosystem. You need dry soil conditions, not bone dry. It needs some moisture, but it needs to be relatively dry during early tuber formation for infection. So this pathogen only infects at a specific time frame. So that should be my little disease pyramid actually. So we've got a time here is critical, our environment is critical. So the time period is when our stolon, our underground stem, is just starting to swell and produce a tuber, and then as it's elongating, we're forming a little young tuber. During that elongation phase is when infection occurs. And why does it occur there? Because the lenticels are fairly immature. They aren't packed full of... in fact, they actually are in the process of converting from stomata to lenticels at the time. So prior to them being absolutely packed full of packing cells, infection occurs then. But also, you're getting these growth cracks as the tuber elongates because it's growing quite fast. And that also provides openings for the pathogen to get in. So it invades during that early tuber infection phase. Once it invades, then disease develops, and at harvest you see horrible spots. We mentioned this before. Absolutely critical for the pathogen to be a pathogen is its capacity to produce a phytotoxin. The phytotoxin is called Taxomin A, that's the chemical structure if you're interested. This phytotoxin is actually really quite toxic to a range of plants. I mean, it does well with potatoes. It'll actually work against any plant that you put it against. It inhibits cell wall division. It inhibits cellulose synthesis and deposition. In fact, there are people that are actually trying to use this now as a broad spectrum herbicide because it's really quite useful in that manner. The pathogen invades, it starts to switch on, it recognises, and there's actually host components, there's host chemicals from the cell wall of the host that actually say, hey, well done, you've actually invaded, and that switches on these genes which switch on production of the toxin. So it knows that I'm now inside a potato, I better switch on my my toxin producing genes and actually start causing infection. And so that switches on the toxin then starts killing cells, but also in advance of that the pathogen invasion, the toxin moves through and starts to initiate cell proliferation. So the plant responds, its defense response is, I'm being attacked, I better start suberizing my cells, suberin, which is a protective waxy compound. So trying to protect the cell and produce these corky layers and actually produce that scab and limit the infection. So that is the formation of the symptom that we see. Oh, sorry, there now. We've kind of said this. So the toxin stimulates suberin, stimulates that protection underneath. That stops the tuber losing. Suberin is also really good at stopping moisture loss. So this is a damage, and it protects the tuber from any further moisture loss. It protects it from additional microbial attack, and protects it from further invasion by the pathogen. Makes it ugly, but actually works really well from the potato's perspective at stopping disease get any worse. As we said with our disease triangle, the environmental factors are absolutely critical. Low soil moisture at tuber elongation is important. That's unusual. Most bacteria like high soil moisture. The main reason this likes low soil moisture, we'll talk about that later in a minute, is that it's a survivor but a very bad competitor. So you have high soil moisture, other bacteria will proliferate and outcompete the pathogen for its ability to reproduce. So this guy survives and does well in low soil moisture environment where other things can't compete that well. It actually likes high soil temperatures. Its optimal growth temperature is about 30 degrees, which is a bit weird when you talk about Tasmanian soils. But it'll obviously tolerate cooler temperatures too. It likes neutral or slightly alkaline soil pH. It likes liming. It doesn't like other bugs. It prefers to have it to itself. So if you've got a really rich soil microflora, you're going to have much less common scab. If you've got a trashed soil that's absolutely hammered, that you've denuded a lot of you've beaten it up, a hard pan or whatever else, a general absence of soil microbial activity, you'll have much more of a disease issue. It does love organic matter as well. Okay, how do we control it? That's a really good question. The main control, probably globally, apart from resistance, is through irrigation. We said before that it needs dry, warm, dry soil conditions around tuber elongation, when these tubers are starting to form. So if you monitor your crop, you can actually predict when that tuber elongation is happening, and you'll know it occurs over about a 4 to 6 week period. So over that 4 to 6 week period, you need to make sure that you've got a good irrigation schedule that will actually keep the soil moist. It's also important, obviously, for development of the tubers anyway. So you've got to counter that with how much water you've got and how the cost of that water. But skimping on your water during that period will actually increase your risk of common scab disease. So again, in Tasmania, particularly with the processing companies, they're encouraging regular scheduling during that period, which has been quite effective at reducing common scab disease incidence. So we know it works, and there's some great data to show it works. As to how it works, there's a few theories, but it might be a combination of all of them. Firstly, this organism is also an obligate aerobe. It needs oxygen. If it's remotely anaerobic, it doesn't grow very well at all. So if you've got moderate amount of soil moisture, things can be slightly anaerobic. That'll inhibit its growth. Number two, it doesn't like competing bacteria. So higher soil moisture, you've got more other bacteria growing, and it's going to struggle to survive. Number 3, we talked about our proliferating lenticel filling cells. If you encourage development of those filling cells and particularly pushing them out and sloughing things off, you're actually making it difficult for the pathogen to invade through those lenticels by proliferation of the filling cells. And the last is that you actually will cool the soil too. So this thing does like warm temperatures. When you irrigate it, you will actually cool the soil as well. So all of those factors are probably important in how irrigation works. We also said it doesn't like acid soils. And a survey around Tasmanian soils showed that anything below about 5.4 disease was uncommon. And as you increase up to 7-ish, disease increases. So that's great. Potatoes are more than happy to tolerate 5 or less. But pH management is still problematic, particularly under rotation. So potatoes can tolerate pH 4.5, but brassicas can't. So you grow your potatoes and then you do a broccoli crop after that. If you have it at acid pH, you're going to get clubroot. So then you've got to toss a huge amount of lime on to try and get the pH back up again. And you're forever adjusting soil pH. And soils, I'm sure Richard Dawes already told you, are fairly robust things. And they're actually pretty hard to change, particularly some of the more robust cropping soils. You can toss huge amounts of sulphur or lime on them, and it can be quite difficult to actually get a big pH boost or crash. What is clear is that liming will increase disease. And there's some argument whether it's a pH effect, it probably is, but also whether calcium also has an effect in there too. So if you've got an acid soil condition and you're actually liming immediately prior to potatoes, you're probably going to increase your risk of common scab quite markedly. And that's very clear. So soil pH, you could perhaps look at acidifying your soil, maybe not the best option, but certainly try and avoid liming if you can. From the pathogen perspective, so that's the environmental perspective. From the pathogen perspective, what can we do? We can try and avoid it as much as possible. So don't plant scabby seed. Seed certification is really critical, so make sure that what you're planting is clean. You can actually put on some seed treatments. Most seed treatments are actually quite effective against the pathogen. So if you buy the best seed you can, the cleanest seed, put on a seed treatment, you're going to reduce the risk of inoculum coming on the seed quite markedly. If you don't, and you do plant scabby seed, you're going to be introducing it and you're going to get a problem if the environment is conducive. In the soil, much more difficult to deal with. This is a great saprophyte, so half of it's coming from the seed, some of it's coming from the seed, a big chunk's coming from the soil. What do we do about that? Can we detect it? The current technologies don't work very well. We can, from historical knowledge of that field, we might know that's a bad paddock for common scab, but it's actually very difficult to find it. Our diagnostics are very, very difficult. So it's hard to avoid. And in reality, the diagnostics we do have, which might not tell you how much you've got, but tell you if it's there. I think if you take any soil sample from anywhere in Tasmania, you're going to get a positive. It's pretty well everywhere. Chemicals, soil chemicals. Look, there were some older chemistries that worked. They're no longer registered. There's nothing really registered in Australia for common scab control. And those that were around, or those that are around, even if they were, are not that effective. Rotation is valuable but not effective and not going to solve your problem, certainly not a silver bullet. This is a great saprophyte, it will survive. The longer you leave it, the more the population will decline, but it won't go away. Biological suppression, that is certainly has potential, and there's work, particularly out of the States, which have shown the value of this.
Before we get into that, I was just wondering, what about soils in pastures which freeze over winter? Because where I come from, traditionally they've grown potatoes and the entire field is frozen up each and kill bugs?
It probably survives freezing as opposed to. I'm thinking probably a better example here are some of the northern US fields where the way they grow potatoes is their rotation is potato and snow. So they grow potatoes and then it gets buried in snow and freezes and they grow potatoes again. They get away with it because most of the pathogens kind of die, but they still can get common scab. It will. I mean, most things don't like freezing, but it probably will survive. I'm trying to think. I mean, it likes hotter conditions. It certainly dries, but I don't think freezing will eliminate it. We can certainly freeze inoculum in the lab and it'll happily survive. But that's probably better than not. That's one of Australia's issues is we generally have too mild a climate as opposed to most other cropping regions, in that some of the extreme heat or extreme cool that might eliminate some of these pathogens doesn't occur. And we usually have everything kind of nice for the plants, but also for the bugs. As I said, it's a poor competitor. We know there are soils that are suppressive and will make it, will eliminate the pathogen. And those soils, they've isolated antagonistic microbes, and these microbes definitely will outcompete the pathogen. And when they've applied them to other soils, they will they'll make those soils antagonistic and make it actually reduce disease. So because it's a bad competitor, you can use biological control, and it does work. The economics, however, are very marginal. There's no product yet that's actually out there that's actually economically feasible to apply to, because you've got to apply a fair amount of it across your entire paddock, and that's quite expensive. So it works, but it's just not economically feasible at this stage. We can get resistance. There are resistance within cultivars. There's nothing that's immune. Stuff that we've been playing with, which is kind of fun. We were doing some work and also following on from stuff that came out of the UK, is that you can actually treat it with hormones, particularly auxin treatments, and that, which are a foliar treatment and yet can control a soil-borne disease, which is kind of a bit weird. But what this does is these auxins and some other chemicals, they actually move through the plant, they get into the tubers, and they actually inhibit the toxin's ability to cause damage in the tuber. So here we go, here's an example, here's our control, here's some ones that have been sprayed with 2,4-D that would normally, that's a herbicide, but put on really dilute levels, and you can get nice clean tubers from that. And it's not registered here in Australia, but they do have it registered in other parts of the world where they're starting to play around with the options for auxin sprays for controlling common scab, amongst other things. Another thing we sort of played with was, again, because we know a toxin's absolutely essential to disease, was that can we develop resistance to the toxin? So we've got our favourite potato, took it into callus culture, individual cells. So we go through tissue culture and we take individual cells. We then threw the toxin at that cells and killed off 99% of everything. Recovered those ones that survived, regenerated them, and thought, okay, are these now resistant? Turns out that most of them weren't toxin tolerant, but a lot of them were disease resistant. And what we've actually selected for was actually plants that actually have much greater ability to produce that defense suberin content, suberin process. So they got tougher skins. So here, for example, is our control under the same conditions with one of our strains, and that might be a minor tiny little lesion there. So it's not immune, but it's pretty well cleaned up. So about 90+% disease control by selecting for mutants that through that process. Now we're going to try and use some of those more modern molecular techniques to also enhance that suberin production, switch off some of the repressor genes, and see if we can make super tough potatoes. Okay, any questions? Good. Round two. crown gall. So a bit more of a traditional, well, a gram-negative rod, so a bit more traditional in terms of morphology as a bacteria, but one, again, that has some very unique and interesting ways of causing disease in plants. It's kind of fun. Crown gall. Disease mostly of woody and herbaceous plants. The members of the Rosaceae are particularly affected, so that's all your fruit trees, your prunus species, your roses, etc. Can all be badly affected by crown gall. In Tasmania, we've got a large number of records of crown gall in a whole range of different crops. The pathogen is Agrobacterium tumefaciens. So it is related to Rhizobia species. It's related to, it's in the same Enterobacteriaceae, so the same broad family as the Escherichia coli and all those sorts of guys. But so Agrobacterium tumefaciens. And what makes it a pathogen is that it has a plasmid. So everyone know what a plasmid is? Anyone not know what a plasmid is? Exactly. So your bacteria's got a single chromosome, but you can have pieces of DNA that exist outside of that chromosome, these extra chromosomal DNA, usually circular, called plasmids. And this plasmid that the pathogen has is called the TI, or tumour-inducing plasmid, and that has all of the genes that are responsible for disease on it. So the presence of that plasmid makes that bacteria a pathogen. Take the plasmid away, it's no longer a pathogen. Gram-negative rod, as I said, part of the Enterobacteriaceae. There is a related pathogen called Agrobacterium rhizogenes, which has a different plasmid. It's the RI plasmid rather than the TI plasmid. What this one does is, instead of inducing galls, it induces a hairy root disease, so where you get proliferation of all these auxiliary shoots, and you get essentially something that looks hairy in the root rather than a gall. But we're not going to talk much more about that. Just to note that exists. There are different biovars, different strain groups of Agrobacterium tumefaciens. I'm not going to worry too much about that. That's again for the taxonomists. There's very little biological significance, except for maybe one that's more commonly associated with grapevines than the others. But and generally, they cause the same disease in the same sort of manner. So what is the disease? A gall, a pretty nasty, horrible gall. The galls usually form either close to, at the soil line or below the soil line. So either in the and in the crown part of the plant, hence crown gall. So it's either just below the soil surface or just above the soil surface. Occasionally they'll grow further down in the roots, but that's usually where the infection occurs. The gall is a hypertrophic growth of undifferentiated plant cells. This is a cancer. So this is essentially plant cancer. So the cells are growing in an undifferentiated manner. They're doing no benefit at all to the plant, and eventually can cause damage, but occasionally can kill the plants too. So very cancer-like for a plant. Yields can be affected, particularly when the trees are young, and very young trees can be killed outright, where these things grow and actually crush the vascular tissue as they're growing. Usually though, older plants will succumb and not be seriously affected. So it's only a real big issue in nurseries and the like. You can get wilting, you can get damage to vascular tissue when the gall grows. The gall has no vascular tissue in it, but if it grows and expands, and it can actually start to crush the vascular tissues. Okay, so let's talk about its pathogenicity cycle. As with common scab, this guy is also a very, very good saprophyte, and it will survive in the soil very well for a long, long period of time. It's commonly found in the root rhizosphere, and also are found in, if you've got a previously infected plant around the outside of that gall, it'll actually quite happily survive there for long periods of time too. So if you've got a contaminated nursery site, then that's always going to be a problem. It's spread from plant to plant by movement of soil. Of course, it lives in the soil, anything that moves the soil around. Also movement of water, particularly soil water, but also movement of propagation material. So if you've got infected transplants, etc., it can also move the pathogen around. We're dealing with a lot of perennial host systems, so of course you're also dealing with pruning, grafting, to make put your root stocks on, your fruiting stocks, so all of that can also transmit and transfer the bacteria if it's not disinfested. Infection. It gets in via wounds. Mostly commercially by cultural practices, but it can also be any other sort of wound sorts. Particularly nasty is when you think about you're a nursery producer and you want to produce lots and lots of apples or lots and lots of cherries, and you want to make them to sell them to orchardists. You've got a different root stock to a fruiting stock. So you've got a root stock that might be a dwarfing root stock, and then you put your different fruiting stock on top. That grafting is a fairly significant wounding site and a really brilliant site for Agrobacterium to invade and start causing problems. So again, that example we had in the prac class, if you remember, I think it was the rose. You could tell it was right at that graft site where the rose had been. You had your rose root stock, and they put they put the flowering variety on top, and at that site, that's where infection had occurred. Plant wounds release certain stress phenolic compounds, particularly acetostringone, which is a stress phenolic, and this material is attractant to the bacteria. So the bacteria lives in the soil. It's actually motile. It's got a flagellum, so it's gram-negative. It's got a lovely flagella. It swims through the soil moisture, and when you've got a wounded plant which is releasing this chemical, it sniffs it and it follows the concentration gradient by chemotaxis, which is movement towards a chemical gradient. So it can sniff the presence of a wound. Quite clever. So rather than just swimming aimlessly around, it goes, oh, there's a wounded root, I'm going, and starts swimming towards it. And it goes from low concentration to high concentration of acetostringone, which directs it to the wound site. The TI plasmid is not essential for that, but enhances the chemotaxic attraction. Obviously, you need free water, otherwise it can't swim. Once it invades, it gets into the apiplast once again and starts to multiply. This is where it gets a bit different. Unlike most plant pathogenic bacteria that sit in the apiplast and go, have a toxin, have an enzyme, and tosses things, this guy gets a bit more intimate, gets a bit more close to its host. So it actually binds to the wounded cell. So the bacteria will swim up, recognise that wounded cell, and actually physically bind to the wounded cell. It's a two-step process. Firstly, it kind of a weak bonding, which says, am I compatible with you? And then decides, okay, well, first date went okay, now we're going to get married, we're going to be bonded permanently. And that actually forms physical bonds between the bacterial cell and the plant cell. And it actually goes through essentially what is the same as conjugation. So conjugation is where two bacterial cells have sex. So for two bacterial cells, they bind, they form bridges between them, and they can pass nucleic acid material between them. Here it's doing the same thing except as a bacteria with a plant cell. A bit kinky, anyway. The genes for adhesion are in the chromosome, so it's not in the TI plasmid. So that actually allows them to bind to the plant cell. Once it's bound to the plant cell, well, as that's happening, that same compound that attracted it in the first place activates genes on the TI plasmid and starts getting them expressed. So the TI plasmid, what those genes do, first off, it actually enhances or encourages cutting out at this left and right border, chopping a piece of the tDNA and excising that from the plasmid. So it cuts off what we call the tDNA region, which contains these auxin, cytokinin, and opine synthesis genes. So these genes with bacterial versions of auxins, bacterial versions of cytokinins. That tDNA is cut out, and then several copies of it are then transferred through, by conjugation essentially, through those pores into the host cell. It then incorporates into the host cell in a random spot and then starts getting expressed. So they've all got efficient promoters, so once they get incorporated into the host cell, they then start expressing those bacterial genes in the host cell. So that is genetic engineering. Genetic engineering is, that's how it works. That's how scientists actually do it as well. So it's a natural process, and this is how this bug does it. So it engineers the plant to express bacterial genes. These genes will produce these synthetic plant hormones, so auxins, cytokinins, and that causes the undifferentiated cell growth. So the plant's all of a sudden been producing all these hormones that say divide, 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 and then these cells start dividing like crazy and producing the gall. The plant can't stop it at that point. It's engineered to do that. You can kill the bacteria at that point, and it still will do it because the cell has been transformed. The cell has got the genes that will force it to produce these hormones. Why does the bacteria do that? Well, that other gene, the opine gene, the opine synthesis gene, is the key. These opines, and there's a whole range of different ones, are specialised amino acids that most organisms can't use. So they're a bit weird amino acids. But the bacteria has a particular catabolism or particular ability to use the opine that it's telling it to make. So what it does is it transforms a cell and says divide like crazy and make this particular weird amino acid that only I can eat. So it's created a food factory essentially. It's made the plant divide, produce this massive gall and produce this stuff that the plant can't use, but gee, guess what I can, and I'm going to do really well, thank you very much. So it takes it very, very clever really from a pathogen perspective. So, okay, disease expression. So once it's transformed, as I said, you could, if you chose to, blitz the plant with antibiotics, completely kill the bacteria. That won't stop the disease because the transformation, the engineering of the plant is irreversible. So it's going to keep going. You're going to keep getting your gall. So you could argue that at this point, the disease is independent of the bacteria. It'll keep growing, cancer-like, divide, forming these galls. It doesn't have vascular tissue, so it doesn't actually have any benefit whatsoever to the plant. It's a complete cost. And the bacteria live around the outside and go, yum, okay, I've created my food factory, I'll live on the outside. They don't live inside the galls, they live around the outside, and these opines are produced and exuded out, and it can proliferate quite happily. You could argue that maybe the bacteria is not really the pathogen, the TI plasmid's the pathogen, and that the TI plasmid is the thing that replicates and actually survives and induces the disease, because you can get rid of the rest of the bacteria and it doesn't stop the disease, but the TI plasmid, or the tDNA I should say, is still actually causing the disease. But anyway, that's by and by. How do we manage it? We can try and avoid inoculum if we target the pathogen. We certainly want to plant clean stock, but at that stage, infection's already occurred. It's just a case of I don't want to apply a diseased plant because it's not going to grow very well. We can try and avoid movement of contaminated soil, but if we know there's a site that's actually got lots of crown gall bacterium in it, then maybe we don't grow, don't use that for a nursery stock. Long rotations will eventually reduce populations, but this is a really good saprophyte. It's hard. Soil fumigation is not that great, not really used. Chemical treatments aren't going to work. Remember, killing the bacteria doesn't stop the gall, so you're talking about prevention rather than curing, so it's not that useful. The environment, we can try and avoid excessive wounding. Maybe insecticides to stop soil, those root-feeding insects. Avoid stem grafting, use budding instead, so getting the grafts further up the stem. But in the end, a lot of that's driven by horticultural practice, that you need to do those grafts. You need to do root trimming. A lot of those, again, your nursery stocks, you pull them out, you trim off the roots because you've got to be able to fit them in a bag to actually take them around, to transplant them. You can't have massive root system to transplant efficiently. So there's a lot of, in handling those particularly in a horticultural perspective, there's a lot of issues with avoiding wounding. This is neat though. This is one of very, very few examples where biological control works for a plant disease. Biocontrol is great for entomology in many cases. It's always, everyone's tried to do it in plant pathology, it generally fails miserably. This is where it works really, really well. And in fact, it's actually by an Australian who really nailed this. It is the main control for this disease, and now really it's not. It's a bit of a non-issue. Crown gall is no longer a significant disease globally. A very closely related species, Agrobacterium radiobacter, which is non-pathogenic, antagonistic strain is what we use. This particular strain is useful because, number one, it's a direct competitor for the ecological niche. It similarly is attracted to that acetostringone. It similarly gets in the same wound sites, and it similarly can bind to that wounded cell. So it actually occupies that infection site and outcompetes the pathogen. It produces a bacteriocin, which is an antibacterial compound, which inhibits the pathogenic Agrobacterium species. It stops DNA synthesis. So it actually specifically inhibits the strains that actually have the TI plasmid. So wonderful, perfectly. It doesn't kill all bacteria; it kills only the pathogenic bacteria. And it also can eat the same, even if infection did occur. It's probably too late at that stage, but it can also catabolize exactly the same opines that the pathogen is, so it can compete with the pathogen for its food too. So it's a triple whammy, but particularly those first two are critical. The bacteriocin, agrocin, is taken up by the pathogenic species, stops DNA synthesis, stops the production of that tDNA, and actually stops the transformation and therefore the engineering. So even if the cells do manage to bind, a few do manage to bind to the infection site, they get switched off. How does it work? The key thing about biological control is, for plant pathogens, is you've got to get the biocontrol agent in before the pathogen gets in. Because once the pathogen invades, it gets inside a sterile environment, inside a plant, and you can't chase it with a biocontrol agent. Because we know most of the wounds occur when you're root pruning or grafting, what they do is they have a bucket of biocontrol agent, they get their plant, they graft it, they dunk it in the bucket of biocontrol agent, then they put it in the soil. So all of the wound sites are already saturated with the biocontrol agent before the pathogen has a chance to go into it. So it's timing. It's getting the biocontrol agent in there first, and because you're handling individual plants, you can actually do that quite efficiently. Interestingly, the worry was the biocontrol agent might acquire the TI plasmid, so they've actually engineered the biocontrol agent so it can't conjugate, so it can't actually acquire the TI plasmid, so it'll remain a biocontrol agent and not become a super pathogen. No resistance, really. Last bit. Time. Last slide. It's nature's genetic engineer. It transforms plants. Where it's been used now is mostly for genetic engineering of plants. So scientists discovered this, they worked this out and went, oh, this is amazing. So all of this genetic engineering stuff is not, the scientists aren't that clever. They're really just, the bacteria is the clever thing. All they do is they take the virulence factors and chop those out and replace it with the genes they want. So we don't want auxins, we don't want cytokine and synthesis genes, we don't want opine synthesis genes. We want a herbicide resistance gene in there. So let's take that rubbish out, we'll put what we want in there, give it back to the bacteria, put it onto the plant, let it do the whole transformation process. I've got an engineered plant. And that is essentially how most genetic engineering occurs these days. Okay, I'm out of time again. Any questions?